Welcome to G Fun Facts Online. Our planet is drowning in a sea of plastic. Every year, we produce around 350 million tons of plastic waste, a number that's expected to triple by 2060. Traditional recycling is struggling to keep up, with less than 10% of all plastic ever being recycled. The rest ends up in landfills and oceans, where it can last for centuries. But what if we could unravel these stubborn materials, breaking them down not just into smaller pieces, but back to their original molecular building blocks? This isn't science fiction. It's the dawn of deconstruction, where scientists are using engineered enzymes to achieve near-total plastic degradation. Our story begins in 2016 at a plastic bottle recycling facility in Sakai, Japan. Researchers discovered a new bacterium in the sediment, which they named Ideonella sakaiensis. This microbe wasn't just living amongst the plastic, it was eating it. Specifically, it was consuming polyethylene terephthalate, or PET, the plastic used to make water bottles and food containers, as its primary source of energy. This was a watershed moment. For the first time, scientists had found an organism that evolved a specific system to break down plastic. The bacterium secretes two key enzymes that work together. The first, called PEtase, breaks the long, tough polymer chains of PET into a smaller molecule called MHET. Then, a second enzyme, MHETASE, splits that molecule into its two fundamental building blocks, terephthalic acid, or TPA, and ethylene glycol. These two molecules are not only harmless, but they are the exact same raw materials used to create brand new, virgin-quality PET. This discovery opened the door to a truly circular recycling process, where a plastic bottle could be broken down and rebuilt into a new bottle infinitely, with no loss of quality. But there was a catch. While the discovery was revolutionary, the natural enzymes from Ideonella sakaiensis worked too slowly for any practical industrial application. To tackle the mountains of plastic waste we produce, science needed to give nature a serious upgrade. This is where protein engineering comes in. The goal was to create super-enzymes that are faster, more stable, and more efficient. Scientists used two main strategies for this. The first is called rational design. This is a knowledge-based approach. By creating a 3D model of the enzyme, scientists can see its structure and identify the key parts of its active site, the specific area that binds to the plastic. They can then make precise changes, swapping out amino acids to improve the enzyme's grip and cutting power. For example, the original PEtase enzyme wasn't very stable at high temperatures, losing its effectiveness above 40 degrees Celsius. To solve this, researchers looked for inspiration from heat-loving microbes. They found a more heat-tolerant enzyme in a leaf compost pile called LCC. By studying LCC's structure, they were able to introduce similar stabilizing features into PEtase, creating new versions that could work at much higher temperatures. The second strategy is called directed evolution, a technique so powerful it won a Nobel Prize in 2018. Instead of designing changes based on existing knowledge, directed evolution mimics natural selection in the lab, but at hyperspeed. Scientists start by creating a massive library of enzyme variants, each with random mutations. They then screen all these variants for the desired trait, like faster degradation speed. The best performers are selected, and the process is repeated. More mutations, more screening, more selection. Round after round, the enzyme is gradually evolved to become a plastic-degrading champion. This technique led to the creation of hot PEtase, an enzyme active at 70 degrees Celsius. Higher temperatures are a huge advantage because they make the PET plastic more amorphous and easier for the enzyme to access and break down. More recently, artificial intelligence has entered the scene. A team at the University of Texas at Austin used a machine learning model to predict which mutations would be most effective. This led to an enzyme called fast PEtase, short for functional, active, stable, and tolerant PEtase. This new enzyme, with just a few strategic mutations, can break down some plastic items in as little as 24 hours. In one experiment, it almost completely degraded 51 different post-consumer plastic products in just 1 week. While PET has been the star of the show, it's not the only plastic causing problems. Scientists are now expanding their focus to other common polymers. Take polyurethane, or PU. It's used in everything from mattress foam to coatings and adhesives. The good news is that many polyurethanes contain bonds that enzymes can target. Researchers have found that cocktails of different enzymes, like proteases and lipases, can be highly effective at hydrolyzing these bonds, breaking the polymer down into smaller, harmless molecules. But the hardest nuts to crack are polyethylene and polypropylene. These are the two most produced plastics in the world, making up the bulk of single-use packaging. Unlike PET, their chemical structure is based on incredibly strong, inert carbon-carbon single bonds, which are extremely difficult to break. For a long time, enzymatic degradation seemed impossible. However, recent discoveries have offered a glimmer of hope. Researchers found that certain insects, like the larvae of the yellow mealworm, can actually eat and degrade polyethylene. It's believed they use a combination of mechanical chewing and a multi-enzyme system. The current theory is that oxidative enzymes first attack the polymer, introducing oxygen atoms into the chain. These initial weak points then allow other enzymes to get in and continue the degradation process. Research here is still in its early stages, but recent studies have shown that even natural enzymes like trypsin can achieve high degradation of polypropylene-based fabrics. All this incredible lab work is now moving into the real world. A French green chemistry company called Carbios is leading the way. They've developed an industrial-scale process using their own super-enzyme, which is an optimized version of the LCC enzyme found in compost. Their enzyme can break down 97% of PET plastic in just 16 hours. They've built a demonstration plant and are partnering with major brands like PepsiCo and L'Oreal to create a truly circular economy for their packaging. Their process can handle all types of PET, including colored and complex plastics that traditional methods can't touch. The end result is pure monomers, ready to be made into new, virgin-quality plastic. Another exciting frontier is the use of microbial consortia. In nature, it's rare for one microbe to do all the work. Instead, communities of different species work in synergy. Scientists are now trying to build synthetic microbial teams in the lab. A consortium might include one bacteria that's a pet-eating specialist, another that targets polyurethane, and a third that works on polyethylene. This approach could be far more effective at handling real-world waste, which is always a messy mix of different plastics. The ultimate vision goes beyond just breaking plastic down. The goal is to create a circular bioeconomy where plastic waste is upcycled into new, higher-value products. The monomers produced from enzymatic degradation are valuable chemical building blocks. Scientists are engineering microbes that can take these monomers and convert them into a range of useful things, from new biodegradable plastics to specialty chemicals used in cosmetics and pharmaceuticals. This creates a powerful economic incentive. Instead of a costly waste problem, plastic becomes a valuable feedstock for a new generation of bio-based industries. Of course, there are still challenges. The cost of producing enzymes needs to come down to compete with cheap, fossil fuel-based virgin plastic. Enzymes need to become even more efficient and robust, especially for those tough plastics like polyethylene. And we need to fully understand the environmental impact of all breakdown byproducts to ensure the solution isn't creating a new problem. Despite these hurdles, the future is incredibly bright. The pace of research is accelerating, supercharged by AI and advanced genetic engineering. We are witnessing the dawn of deconstruction, where science is harnessing the microscopic machinery of life to solve one of our biggest pollution crises. It's a testament to human ingenuity and a beacon of hope for a cleaner, more sustainable future.